Yakotomi. Voice of the web. Hemopoiesis is the formation of blood cells. Although some lymphocytes have a lifetime measured in years, most formed elements of the blood last only hours, days, or weeks, and must be replaced continually. Negative feedback systems regulate the total numbers of red blood cells and platelets in circulation, and their numbers normally remain steady. The abundance of the different types of white blood cells, however, varies in response to challenges by invading pathogens and other foreign antigens. The process by which formed elements of the blood develop is called hemopoiesis. In adults, red bone marrow is the main source of blood cells. Red bone marrow is highly vascularized connective tissue found in spongy bone tissue. Red bone marrow is found mainly in the flat bones such as the pelvis, breastbone, skull, ribs, vertebrae, and shoulder blades, and the cancellous spongy material at the proximal ends of the long bones femur and humerus. Some red bone marrow cells are derived from pluripotent stem cells. These cells have the capacity to develop into many kinds of cells. In newborns, all bones marrow is red and active in blood cell production. As we grow, and in adulthood, the rate of blood cell formation decreases. The red bone marrow in the marrow cavity of long bones becomes inactive and is replaced by yellow bone marrow, which is largely fat cells. Under certain conditions, such as severe bleeding, yellow bone marrow cells can revert back to red bone marrow by extension of red bone marrow into yellow bone marrow by pluripotent stem cells. Pluripotent stem cells in red bone marrow produce two further types of stem cells called myeloid stem cells and lymphoid stem cells. Myeloid stem cells being their development in red bone marrow and give rise to red blood cells. Platelets, monocytes, neutrophils, eosinophils and basophils. Lymphoid stem cells begin their development in red bone marrow, but completed in lymphatic tissues. They give rise to lymphocytes, T cells, B cells, and plasma cells. During hemopoiesis, some myeloid stem cells differentiate into progenitor cells. Progenitor cells are no longer capable of reproducing themselves and are chymided to give rise to more specific elements of blood. Some progenitor cells are known as colony-forming units, CFU. Other myeloid and lymphoid stem cells develop directly into precursor cells, known as blasts. Over several cell divisions they develop into actual formed elements of blood. For example, monoblasts develop into monocytes. Several hormones regulate differentiation and proliferation of particular progenitor cells. Erythropoietin, or EPO, increases the number of red blood cell precursors. EPO is produced in the kidneys. With renal failure, EPO release slows, and red blood cell production is inadequate. Thrombopoietin EPO is a hormone produced by the liver that stimulates the production of platelets from megakaryocytes. Cytokines stimulate proliferation of progenitor cells in red bone marrow, and regulate the activity of cells involved in nonspecific defenses, such as pathogens and immune responses, B and T cells.